Hey everyone, we go ahead and get started. Thanks for coming. I know it was late notice, but hopefully it can be helpful. So yeah, we'll just go ahead and get started. Okay, so just a brief overview. This is just all of the lectures and pre-lecture topics that you guys will need to know for the exam. It is a lot of information, but hopefully you guys have been studying for all the quizzes, so it's kind of all up there. You just gotta pull it out. And then that's also like a picture from the homepage of the Canvas site with all the links to everything. And this review session, these slides, and the video that we're taking will be up next to the exam one with slides and videos thing. And then just to quickly go over the format of the test again, it's going to be almost exactly the same as all the quizzes, just 4 times the length. The questions are all going to be very similar types of questions. They're all in the same question bank. So it's literally just going to be the quiz times 4 and on all of this material instead of just 2 topics. So we'll just go ahead and, actually, sorry, is there going to be questions? And that will include the material from today, so make sure you're studying that as well. Okay, so first off, we have the amino acids pre-lecture. The most important things to know from this are the amino acid basics, so the structure of like all general amino acids. So just know that there's an alpha carbon, and then attached is an acidic carboxylic acid group, a basic amine group, and then a side group or residue. And that can be different for every amino acid. And you don't need to know, you don't have to have the side groups structures memorized. And then also that peptides are read from the N terminus to the C terminus and connected to peptide bonds. And so you should have the amino acid names and groups that they fall under memorized, as well as their 3-letter abbreviation. But you don't have to know the specific structures. Unless as you go through the slide, there's like a specific amino acid that we talk about in relation to a specific mechanism or disease. Okay, and then from the lecture, again, we're really focusing on peptide structures here. So it's really important for everyone to know the hierarchies of structure. So you first have the primary structure, and that's just the string of amino acids, and you again read that from the N terminus to the C terminus. Then you have the secondary structure, so those are the alpha, helices, and beta sheets, and that's driven by hydrogen bonds that cause local folding. Then you have the tertiary structure, which is the folding of individual subunits. And then you have the quaternary structure, which is assembly of multiple subunits. So there will be multiple tertiary structures in a quaternary structure. And then it's really important to, as you go through and study, make sure you know what bonds determine each level of structure. So for example, disulfide, ionic, and hydrogen bonds, as well as hydrophobic interactions, are all very important things when it comes to determining different levels of structure. And then also, you don't have to know anything about like symptoms or etiology of sickle cell anemia, but you should know like the applications of amino acids and peptide structures and how that applies to sickle cell. Okay, and then just wanted to give you guys a little bit more practice with reading peptide chain, because that will likely be one of the questions on the test. So again, you can read peptide chains from the N terminus to the C terminus, and it won't always be given from left to right. But the easy way to remember that is you find the like amine group all the way on the left side, and you look to the right, and if there's an alpha carbon there, so the alpha carbon is the carbon that has the side group attached to it. So sometimes they'll have the actual side group, sometimes it'll just be an R. And if you look, so when you find that amine group, the direction that you go to find the alpha carbon is the direction that you read the peptide. Does that make sense? Okay. And then a little practice with that. Can anyone tell me? Okay, well, we're kind of giving it away, but just again, you find the amine all the way on the left, and then if you look to both sides to see where the up carbon is, whatever direction it is where is the direction that you read the peptide in. So definitely know this. I know it can be a little bit hard, but this will definitely be on the test.
Okay, so next we have the membranes and cell structure pre-lecture. And so, as you guys know, you should know the different organelles and what their functions are, especially those that have multiple functions. And then as well, the secretory pathway of the stuff out of the cell, so these vesicles as they exocytose out of the cell. Be mindful of the reducing environment of the cytoplasm versus the oxidizing environment of the organelles which are so place. We talked about this in the lecture. If you have any questions with that, there's a pretty good slide on it in that lecture. And basic scaling, this was a big thing, right? There's a slide in the pre-lecture about this. Basically, just know the scale of the cell compared to organelles and other components of like the sublamina, that kind of thing, because that could possibly show up on the test. Okay, so now in the main lecture, some of the important things, the composition of the cell membrane. So right, we have the fluid mosaic model, that lipid bilayer, and also the proteins that run through it. So the proteins associated with the cell membrane, the peripheral, the lipid anchored, and the integral and transmembrane. Right, so those are the four important ones. And then know about, right, the alpha helices are the ones that are used to cross the cell membrane. Definitely know why that is. And then hydrophobicity, right? We talked about these. I think we have one here, right? Yeah, we'll go over that. The membrane functions, right? Transport barrier and also regulation, signaling, and adhesion. Right, the fluid mosaic model, which is in membrane fluidity. Big thing from this that we talked about was cholesterol. And then another thing, the trans versus cis double bonds for unsaturated fatty acids, which one's going to lead to more fluidity versus less. When you have the trans, right, it's going to act more like just a saturated fatty acid rather than unsaturated, while the cis leads to that greater amount of stickiness. And then the phosphotidylserine and the membrane asymmetry. And then how does this come into play in lupus? So we talked about right at the very end of that lecture. So here's that hydrophobicity plot we're talking about, right? Look at how whenever you have a cross here from more hydrophobic to less hydrophobic, you know that's where there's going to be a protein crossing the membrane. So I think this ended up being a quiz question, I think, right? Where you count the number here, and that's going to give you the number of times that you're crossing that cell membrane. So this is definitely, we really harped on this in lecture, and we're going to talk about it again here, right? This is something that you should definitely know. Yeah.
You said it was a membrane protein?
Yeah, it says crossing the membrane.
So hydrophobicity plots are basically the way that you can know how many times a transmembrane protein is crossing the membrane. So in this case, it would be 4 times because there's those 4 peaks. And you wouldn't count like those tiny little ones all the way on the left, and like in the middle there, you just count those bigger ones. All right, so for the ECM, I decided not to split this up into pre-lecture and lecture because it's all interconnected. But we have all these mental things to know. This is where we start getting into realizing, how in detail we go. But again, hopefully you all have been keeping up with the quizzes, studying as we go. And if you haven't, would definitely recommend that going into the next exam, just because it's a lot of information. So the extracellular matrix is a hydrogel that forms its matrix outside the cells. So it being a hydrogel, it means it's filled with water, as well as its other structural fibers, hydrogen matrix, and adhesion molecules. So the structural fibers, you should know the structure and the function. So that's collagen and elastin, and we'll go over those in the next slide as well. Then the things that make up the hydrogen matrix are also the proteoglycans and water, and we'll go over proteoglycans in the next slide. And then we have the adhesion molecules, And those mediate interactions between the extracellular matrix components and the cells. And then they also act as receptors between the cell and the ECM components that serve a structural role and also do some signaling. So the extracellular matrix is split up into two. We have the connective tissue and the basement membrane. So the connective tissue is just in between all cells. It's a collection of proteins and carbohydrates produced by cells, and that forms a matrix outside the cells. And within the connective tissue, you have fibroblasts, which are the cells, fibrous proteins, glycosaminoglycans or GAGs, and proteoglycans, which are made up of GAGs with a protein core. And then there's also some other cells floating around in the connective tissue. You don't need to know that, but there's also immune cells, some other random ones that just float around. And then you have the basement membrane. So this is more like a sheet rather than like a sponge, I guess. And this is under all epithelial and endothelial cell layers. So that's basically any cell layer that's like lining organs or like your skin. And so this is made up of mostly collagen type 4 for tensile strength, as well as laminin, which organizes the basement membrane, and then nitrogen and protein, which form bridges. And we'll talk about some of those things a little bit more in the next slide. So collagen is formed from parallel fibrils. So you can see that image right there, kind of like the breakdown of how it's formed. And it has a really high elastic modulus. And for those of you who haven't taken biomechanics, that just means that when you stretch it, it doesn't have a lot of ability to go back into its original position. So this is mostly used for tensile strength rather than like stretchability, which is not a word, but and then it's really important to know that there are two post-translational modifications to amino acids that happen in collagen to stabilize the triple helix. So those are two amino acids that it might be helpful to know the structure to kind of, or it's one amino acid, but it'll be helpful to know the structure. And then you should also know which enzymes are involved. And then, so I'm giving you, so collagen is stabilized by cross-links between the lysines, and that kind of forms a matrix. And lysines are modified by lysyl oxidase to be able to form those cross-links. So then we have elastin, which is tropoelastin cross-linked, again by lysyl oxidase to form elastin. And it has a much lower elastic modulus than collagen, which means it's more like a rubber band able to get back into its original form when stretched. And it's classified as an entropic spring, so it's important to know that. I'm not going to go over exactly what that is in all the slides. And then you have the proteoglycans, which again are the core protein, and the GAGs branching off of it. And it's really important to know that the GAGs are hydrophilic, so they spring away from each other to form a hydrated matrix, so the water goes in between that matrix. And then they also have the extracellular fluid, and this is all in the lecture. You should know the properties of that extracellular fluid. Last slide on the ECM. So another really important component of the ECM is fibronectin, and this serves a lot of different purposes. So it's kind of called the biological hub. Definitely know its general structure and function. Then we have adhesion molecules, which mediate interactions between the extracellular matrix and the cells. Again, the receptors that serve a structural role. And they also are involved in signaling with the cell about the stiffness of the extracellular matrix. So specifically, we have integrins, which are cell surface proteins on the surface of regular cells that interact with the extracellular matrix. So like we were talking about transmembrane proteins before, integrin are transmembrane proteins, so they have intracellular and extracellular components. But they also have alpha and beta subunits, as you can see in that picture over there. And they serve as mechanical support by connecting to the ECM and kind of anchoring the cells in place. Or they also serve as a signaling platform for cells, again, when the integrins are binding with the extracellular matrix, and providing that support. There's mechanosensing that will tell the cells how stiff that extracellular matrix is, which will affect different cellular processes, proliferation, stuff like that. And then briefly, we also have the cell-to-cell adhesion receptors to know about. So there's just 4 listed there. Make sure you know their general properties and structure. And then lastly, we have the fibroblasts, and these are the cells that reside in the connective tissue, but they're also the cells that secrete the ECM components. And in the case of injury or inflammation, these fibroblasts are activated into myofibroblasts, and they'll go to the site of the injury, and then perform their ECM deposition. And these fibroblasts play a huge role in IPF, like we talked about in the lecture. So that would be really important to know.
So next, we're gonna move into the enzyme-free lecture. So
Actually, wait, sorry.
Yeah.
We just talked a lot of information, you guys. Does anyone have questions about any of that? We'll also have time for questions at the end, but if you have something urgent.
Okay, so next, the enzyme-free lecture. Good to know the basic qualities and categories of enzymes, like specificity, how it occurs, the active site or catalytic site. And they all end in A's, and some important ones, kinases, proteases, peptidases, probably also phosphatases, another good one to know there. The basic mechanism of how enzymes work, so, kind of like those, use for energy diagrams, how they lower the activation energy of a reaction. And then considerations for the optimal enzyme activity, obviously these are names right here, temperature, pH, and then cofactors, coenzymes that are necessary for them to perform that lowering of the activation energy. Michaelis-Menten kinetics, definitely know the equation for it. You don't need to know the whole derivation, definitely not. And then also like being able to find the Km when you are given the Vmax is a good thing to know. We did that in class, right? You're given that table, and once it starts to plateau, then you go back to half that and find whatever the Km was. Then basic knowledge of colorimetric techniques and the unit definition, and then we're going to talk about some more next slide, the turnover rate. You should know how to find the given Vmax and weight. So that's on the slide. Yeah, sorry.
For the Michaelis-Menten equation, you should definitely have this memorized. I know it's like easy to be like, oh, like it's okay, it's just an equation. Definitely have it memorized because it will help you with questions about like inhibition, stuff like that.
And then for turnover rate, the rate saturation for one enzyme molecule. So it's, if you think about it, it's constantly logically, it's how many reactions are catalyzed per second per molecule. And so this is the example we did in class.
It's pretty much just basic unit conversion. You just have to be able to to cancel out everything except for seconds, or 1 over seconds. So this is just an example, but there might be a situation where you're given the Vmax and the molecular weight, and these are kind of steps of how you would get to 1 over seconds, which I know you guys all took chemistry, so hopefully this is intuitive.
Okay, and then into the actual lecture, we talked a lot about competitive versus non-competitive inhibitors. So an ideal competitive is going to alter your Km, but it's not going to change your Vmax, and that's because it's competing to find the active site. And then non-competitive, this is your allosteric binding, and so it's going to alter Vmax, but ideally it will have no change to the Km value. And so it's going to induce that conformational change of the active site from a different location. And so definitely know the difference between the regulatory and catalytic domains. And then domains versus subunits. Domain, different areas of single protein, subunits are multiple proteins. They're a subunit when they become a quaternary structure, so it's like multiple proteins coming together. Different types of regulation, allosteric, covalent modification, subunit association or dissociation, and then cooperativity. I know we kind of talked about cooperativity a decent amount, there's a good slide on that in the lecture, so. And then onto botulinum toxin, it's an enzyme, so it's acting on these B stairs, T stairs, and then the synaptic retina, this is what it's cutting. So it gets endocytosed into the motor neurons due to this receptor binding domain it has. And then the translocation domain pops the lysosome that it's entered the motor neuron in. And then the catalytic domain is free to cleave synaptic retina inside that motor neuron. So that leads to having no vesicle fusion, and then there's no neurotransmitter release, and that's why you get that paralysis effect.
Next up, we have, or actually, sorry, does anyone have any questions about anything related to enzymes before we move on? Yeah.
Can you say how, like, the thing you just said, like again, but slower?
Yeah. Well, I can try. So, okay, V stairs and T stairs are like cell surface receptors that bring in vesicles that are carrying neurotransmitters, specifically on neurons. And synaptogrelin is just a type, I think, of the V stair. So basically the way botulinum toxin works is it enters into a motor neuron cell through endocytosis, so it'll be in a vesicle. And then that is because of the receptor binding domain, it's an enzyme that like makes that happen. Then the middle domain, which is the translocation domain, that basically pops the lysosome. So instead of like eating up the enzyme because it is a toxin, that the toxin escapes and then it's free to do whatever it wants in the cell. So the catalytic domain basically is like a drug sniffing dog that will find the synaptogrelin on the it will find the synaptogrelin part within the cell, and then it will go to that synaptogrelin site and cleave it so that the cell no longer has synaptogrelin and won't be able to uptake any more vesicles, whether that's a lysosome or something it wants to destroy or any other type of vesicle. Which means that the neurotransmitters won't be able to enter the cell through endocytosis and perform whatever functions they would perform, which causes that paralysis. Was that slow enough?
Yeah.
I know this is like a lot, and it's also like you could probably find like another video on YouTube with better graphics, but the most important thing to know about the botulinum toxin is that it's an enzyme with multiple domains that each do different things. That's like the main purpose of learning about the botulism. Any other questions? Okay, so on to the cytoskeleton. So there are 3 different types of like things that make up the cytoskeleton. So first we have the intermediate filaments, and these are non-polars. They don't have directionality like a lot of the other ones we talked about. They don't have positive end and negative end. And these are the most stable, and they're mostly strengthening and stabilizing. And you can see an image in the top right of what they would look like. Then next up we have microtubules. Those are used for intracellular transport, bending, stability, and motility. And those are made of alpha and beta tubulin, and microtubules are a GTPase, so an enzyme that forms GTP, or uses GTP, sorry. So the formation of that is that it will form protofilaments that will then roll into tubes that look like pipes, and you can see it in the image here. It kind of like coils together to make that tube. And these microtubules do have polarity. So this we talked about the end tops in lecture, and I would definitely like go back to the slide because it's going to be important to know how microtubules are formed and to know how like the GTP cap works and all that. But there is like a negative end that stems from the end tops, and then the positive end, which is where the growth takes place. And this isn't all the information you need to know about microtubules, just a little bit of background. Then we have the microfilaments or actin filaments. And these are for contraction and cell shape. And these actin filaments form from monomers and they're in double helix shape. And these are ATPs. So it's important to remember microtubules are GTPs and microfilament or actin filaments are ATPs. Easy way to remember. Okay, so it will be important to know for the microtubules, the phases of polymerization and the critical concentration, to know when there will be growth or shrinkage of those microtubules, as well as like the dynamic instability, and how the GTPase activity is affecting that critical concentration. And then it'll also be important to know the function of microtubules in mitosis and how that's used in chemotherapy. So just to briefly go over that again, the microtubules are basically what's pulling the chromosomes apart. And then it'll be important to remember how actin polymerization works as well, and remembering the characteristics of the positive and negative end, as well as how that treadmilling works. And then we also going into the disease application for that, there are methods of controlling actin polymerization, like certain poisons that affect the cytoskeleton. So in the case of Listeria, it'll be important to know how the ARP-T3 and WASP utilize the actin proteins to enhance its invasiveness, basically like giving it a propeller on its tail to like penetrate through the cell membrane. But that's all in the slides. We're not going to go too in depth about that. Okay, any questions? Oh yeah.
For the actin polymerization by treadmilling, you just mean the addition of
Yeah. Yeah, and just like the mechanism of how that works. Yeah. And that like keyword will be in the slides as well for you to like find where you're studying that.
I know, I just have one clarifying thing on here, because this doesn't get last year, no, WASP and VASP are not the same thing. I remember the first time I saw that, I totally thought this was just like that or something, different things, good to know. But yeah, no other questions, we'll go into the molecular motors pre-lecture. So the basic mechanism of motion for how this working. So you got hydrolysis of ATP, which is causing shape change, and then it's going to bind to these cytosolic filaments. So these microtubule-based motors are like kinesin and dynein. Definitely remember the directionality. It's a good thing. Definitely to know this that the kinesin is the positive and the dynein is the negative. And then they have a motor head and a carter tail, and they're being used for transform. Sorry. So, and then the actin-based motors are the myosins. They also have directionality. They go plus N. They can either, these motor heads and tails are defining cardio or they're forming a filament. We talked about that again today in class. And they are sometimes recessive. And then also just generally, so have some basic knowledge about the ways that we can measure molecular motors.
And then something that helps me remember about the recessive is for the microtubule-based motors. They're basically walking along like the filament. So there's always like one side of the head is always attached to the filament, the cytoskeletal filament, whereas when something's not recessive, it can attach and then detach. But like with recessivity, there's always part of the molecular motor that is attached to the cytoskeletal filament.
On to the lecture, if we know why these molecular motors are necessary for honestly most bodily functions, pretty much just the transport of cargo inside to outside of the cell. The microtubule motors have two domains, as I said earlier, right? They have the motor head and the cargo tail. Know the structures and components of each part. So, talking about the dyneins more specifically, dynein 1. So cytoplasmic cargo transport, dynein 2 is the ciliary flagella cargo transport. And so, yeah,
It's just that there's 3 types, and they all do different things. You should just know the general things for cargo transport and for ciliary flagella.
and then, okay, so these kinds, there are a bunch of different types for the different cargo types. And then the binding of the cargo to the tail is gonna cause a conformational change in the head of the kinesis. And so for rabies, how this works is it's hijacking the dynein and it's transporting it, it's getting it transported to the nucleus because that dynein is going towards the negative.
Yeah.
And then it's sorry.
Oh, it's just like, you can use that to your advantage to do like tracking within the cells. So like you can attach like fluorescence molecules to those dynakinesins to track like cargo or other things. So like in rabies, it's really bad that a virus is hijacking the dynein, but for like experimental uses, it could actually be to your advantage.
Yeah. Okay, and then this, hopefully this is really fresh for you guys since we just did it today, but the active motors lecture. So myosins, positive, the head is the motor domain, and then they also have the tail, which again is connecting to that part of their transport.
Yeah, and to clarify,
Yeah.
for the myosin, it looks a lot different than the microtubule-based motors for it to be like positively oriented. For the like dyneins and kinesis, it basically means that it's like walking in the direction of the positive or negative charge. But for the myosins, it means that it's gonna be basically like pulling two positives together rather than pulling two positives apart, which would mean it would be negative. So the positively oriented means like when we talk about, we'll talk about this with like the cross-bridge cycle, but basically that cross-bridge cycle, because myosins are positive, it's pulling together two positive parts of like the cytoskeleton. Yeah. Rather than the myosin moving towards a positive.
Yeah, it's more about the association than it is about the actual movement in a direction. But we'll talk about it when we.
Yeah, that will make more sense when we
Yeah.
get to it.
And so, right, these myosins, we talked about them mostly as forming the filaments in a sarcomere. And they're also responsible for muscle contraction and then track switching and cartilage, which we didn't talk about as much. Most of those on the muscle contraction thing.
Yeah, you basically just have to know that that's a thing that exists. And like, you don't have to know too much in detail about it, but you just have to know that they're kind of like a back and forth about like which cytoskeleton aspect the cargo is gonna be traveling along.
Yeah, and then so these mechanisms for activation and inhibition of myosins. So you have the actin tropomyosin complex of striatal muscles. Know both theories for how this is happening, right? There's the role that calcium plays, and it talks about them pretty well in the lemma slides. You can go back and get the pressure on the two. And then definitely know the three parts of the trophin complex. And they're obviously really easy to remember because the first letter tells you exactly what it's doing. Right? The I and then the C and the yes and the T. And so the phosphorylation of the regulatory chain in smooth muscle, that leads to activation of myosin, and it relies on calcium to some extent. That's why when you're depleted calcium, it totally stops working. And then it requires both a kinase and a phosphatase because you're both removing and then adding a phosphate group back to this ADP. And so the actin-myosin cross-bridge cycle, first you start and this is right now we're in you start down here and you start in rigor and then you have this ADP which is and then the ADP you're in rigor and the ADP will detach and now you're having an ATP come in and it's going to bind to this component and now you're into this weak association and so this is why you're able to have this back and forth here, which is we'll talk about later is why that which is why we connected it to heart failure originally and that drug. But you're in this weak association, and so ATP will bind and then release from actin, and the ATP hydrolysis will cop back the motor domain, and you'll have the power restricted. So that's what's going to contract. It's going to move it's going to slowly move it closer and closer together, and you're gonna get the sarcomere contracting. And so what happens in heart failure is these molecular motors are not being activated. And so that's why you're not getting as much as big of stroke volume, and so you're decreasing your total cardiac output.
And that could be due to either depletion of calcium ion or lack of ATP. It can also just be due to damage to some general, like other cells. Yeah, and I would definitely recommend going and watching a video of the cross bridge cycle, because I think it can sometimes be hard to visualize. But you kind of think of like a tug of war. But definitely go watch a video, because that will help you visualize it all happening in one process rather than like different snapshots of it. That definitely helps me get familiar with it. And that's it. That's all that we have prepared for you. So I know we didn't go super in depth about everything, but we just wanted to give you a pretty good background of the things that you guys will be responsible for going back into the lectures or into your notes and studying for the exam. But now we'll just have like 25 minutes for questions. We can end a little bit early, but we can take any questions, and then also if anyone has any like individual questions about the hypothesis. Yeah.
Do you guys by any chance have like any questions that you could ask us regarding like the practice questions or something like that other than the quiz?
We haven't prepared any for you guys, so sorry about that. But like Dr. Agabayu and Kenzie are saying, a really good way for you guys to study is to make those questions on your own. As important as being able to answer questions is, like you guys definitely should be developing the skills for the exams to build those connections on your own. But for the next review session, we will discuss the questions.
Are you guys gonna share this slideshow with us?
Yeah, so it's actually already up on the Canvas site right now. If you go to like the homepage where the regular lectures are every week, it's just next to the exam one instead of.
Do you have any advice for like answering the questions where there could be more than one answer, or maybe all the answers, or maybe none of the answers? Like, like we learned from one, but then there's like two others and we don't really.
Yeah. So, as important as it is to use your own reasoning and logic, a really reliable thing is going to be to see how these like explanations were worded from the teaching team or in the lecture slides. This is like, it's hard, but it's really important to pay attention to the detail as you're studying. So what I found helpful is kind of like working from bigger picture, making sure I understood how the thing worked in general, and then kind of working down to more specific parts of the mechanism. And honestly, once you have a pretty good understanding of the mechanisms, answering those kind of questions will come more naturally. Yeah, that's like the best answer that I can give you. Unfortunately, those questions are a little bit meant to, not to trick you, but just to make you like really think back to how the material was taught to you. But I would also just say like don't think too hard about it. And when you have the kind of questions with like multiple answers could be right, try to think back to how stuff was worded in the lecture. Because that's usually, or that will always be what we base things off of, or if it's not, and the lecture said that, then we'll give points back if questions were worded bad, like with quiz trees.
Specifically the lecture or like the lecture slides? Like, do we have to like come through every single lecture?
No.
Or like
Everything, like I would say 95% of everything that's gonna be on the test is written out in the lecture slides. There is some stuff that is like said in between that will help you understand what's on the lecture slides, but I would say you can pretty much base like your answers to the questions off the lecture slides. So definitely be referring back to those as you study. Even if you have your own notes written out, it's still helpful to go back to the lecture slides to make sure you have the correct understanding as you're speaking. I don't know.
Yeah, I mean.
Better answer.
No, I just, on that, they usually stick to the blue terms pretty well, and then if it's not a blue term, it's something they talked about after, like, saying the blue term, it's like, so the definition or something about it. So usually you can get to it pretty well from whichever terms he's already highlighted for you in the lecture, I would say.
I'm just wondering, this is like a content thing, but. So we went over today, like the difference between the classic theory and the newer theory, but I'm not like fully actually understanding.
Yeah, I actually,
Sorry.
I'll pull
pull those up.
I'll pull up the lecture, that lecture real quick, so I can give you a better answer.
Yeah, which that's a great question.
I think also the newer theory is definitely, you can know the newer theory is the one that you should know better, because this is the one, that's the one.
Basically, anytime there's an old theory and a new theory, you should know the new theory better.
The old theory,
PI works, how it's
yeah,
like,
I feel.
allosteric by TNT, which dividing by
Yeah.
Now by.
Sorry, do you have the other question while we're working on that one? Yeah.
You go on the last slide.
Yeah.
I just had a question about so what causes the myosin to like reattach to actin? Is it the like ATP turning into ADP? Is that what makes it like reattach or?
Like in this set, like the top.
Or like the top part, like the circle to the.
Yeah, it's, I would say it's because it's energetically favorable for that to happen. So like, even though it's still weak dissociation, it's going to reconnect because it's in that composition now, and then it's gonna pull. So it's energetically favorable for that.
So like once the phosphorus comes off.
Yeah.
Then it's.
And then, yeah, so like, it's gonna come back and then, right, so you already have the ADP and then you have clock that's gonna come off. So it reattaches. It's because it's still right there, right? It's not like it's associated far away, so it's still right there and there's movement and it just reattaches. I think the best way to use that it's, there's still, it's still, even though it's weak, it still wants to associate with it, so it's just a detach it while it's copying.
Yeah, also like another way to think about it is like when the phosphate group is cleaved from the ATP or the ADP, sorry, no, the ATP. Geez. That's releasing a lot of energy that can be used to cause the bond to be formed between like the myosin and that. Okay.
Okay, yeah. I think so. The difference is that, whenever you have in classic theory, it's saying like whenever you have this conformational change, it's actively that change is transitive, which means it's like signaling to the other domains to move tropomyosin off the myosin binding site. And so the reason that's different is because this newer theory is saying that the inhibitory subunit domain, is locking tropomyosin to actin, and then when you get that, it's just release it. So rather than moving the whole complex off, it's just like opening up the active sites of stuff to bind to it. It's it's not a huge difference, but just in the mechanism of the newer model, it makes a lot more sense. That's why I'm saying focus more on the model, because that's the mechanism we're really talking about more, I would say.
Yeah, I would say just like know that there's two, is probably more important than knowing what the original one was. And also again, like looking at video, it's really hard to conceptualize the crossbridge cycle by just hearing about it and reading about it. Definitely watch a video, because that way you will be able to see how the calcium influx causes the troponin complex to change conformation, which then initiates that crossbridge cycle. And it also helps you kind of understand why the power stroke is happening, why it's going into that cock position, and like how that cycle works. Yeah. Any other questions?
Could we go over like the listeria disease process, like as it like, relates to like 2-3 moss?
But your question is how it's related, like what.
Yeah, I think I'm just a little confused about like, or what R23 is doing. I think I like thought that R23 was like on the listeria molecule or listeria bacteria. Or,
So-
I don't know.
Yeah, so you're right, it is, as you can see in the picture, it is attached to bacteria. What's happening is it's like recruiting the proteins, right? Because it's being active, the RQ3 is being activated, so it's recruiting actin. And then the branching that we talked about, that's happening, and that's how it's moving, it's continuously recruiting actin and making it branch. And that's why it leaves that trail of actin behind it, because that branching is what's propelling it forward, the movement. Oh, it's basically, it's making all those active branches and it's moving away from that. So that's why you're getting that active trail behind it, and that's how it's utilizing the RQ3.
And is WASP involved at all, or?
What? Activate R2. So it's just like, it's part of the cascade, like a signaling cascade. So WASP is just what will activate that R23 cause the actin to form new branch, or to form those branches.
Actually, I think in Listeria, it's binding it and activating it.
Yeah.
Sorry, in Listeria, WASP is not here, sorry. WASP is in the normal like inside the cell, you have WASP that's activating it. Here, when the R23 binds to the bacterium, it's both binding it and activating it.
Okay, gotcha. And so it doesn't have like an RQ3 molecule.
So the RQ3 is here, but there's just no WASP involved in the Listeria one.
So we have like the R23 would always be there, like it just needs to be activated by something.
Oh,
So it, like, a protein can exist on the surface of the cell, and it just wouldn't be doing anything, it's just there, kind of like in its like dormant state, and then something would come in and activate it, so it actually do what it's meant to.
okay, yeah, I think I was just confused because I think the quiz question was like it like recruits and activates. So
Oh, yeah.
Yeah, so that, yeah.
okay, so it isn't on the molecule to begin with, or I guess like what does it mean to recruit a molecule?
Okay, so yeah, so no, you're fine, I understand what you're doing now. So, right, what it means by recruit is the R23 is gonna show up and it can bind, like that active site that it has, it's substrate specific, but it's recruiting because that R23 can bind there, so it's being recruited like it can be out there. So it's recruiting it and then it's also activating, is what's happening, yeah.
I saw him.
Can I ask about the, I guess like the connection between the different troponins complexes and then the phosphatic cycle, like what was the connection? So that like it didn't that cause the contribution?
Yeah, so basically, when they're.
I'm just looking for
Oh,
it.
yeah. Okay, so basically, okay, you can kind of see it up in the image over there. Basically, what happens is there will be a calcium influx into the cell, to the location. And then the troponin C subunit, the calcium is gonna bind to that subunit. And that is gonna serve as the allosteric site. So when the calcium binds to the troponin C subunit, the conformation of that subunit is gonna change, or of the troponin complex is gonna change. So that instead of the inhibitory subunit being the one connected to the actin filament, the tropomyosin binding subunit will bind to the actin filament. And that will allow for the like cross-bridge cycle to So you can kind of think of it as being like, there's not really a beginning because it's a cycle, but it's like the cycle can't start until there's that influx of calcium and the conformational change of the troponin complex on that.
Okay, so like the troponin complex thing like happens before the growth cycle ever happens, so that allows.
Yeah, you can kind of think of it more like calcium will come into the cell. Like, I guess, okay, if you're not moving, nothing's happening, then you can move, calcium will have an influx into the cell, the cross bridge cycle will start, but like, the calcium will eventually dissociate from the troponin complex and it'll return to like its original conformation, but then like I don't know, nanoseconds later, more calcium ions can make their way over to the troponin complex and the whole thing can start again, which like is happening 99% of the time, because like your muscles are almost always moving. Do you
No, that's pretty much what I would say, right? The inhibitory part is locked on, the calcium comes in, it unlocks it, and then the other binding site opens up.
And again, I know I've said this like a million times, watching a video helps so much because it's so hard to explain, and like you can't really like recreate it with anything. Watch a video and like then when you go back and look at the notes, you'll be like, oh, like that's what that means.
The pictures don't do it justice. I don't know. Yeah.
Okay,
But
In the slide, it says like the secondary structure are referred to proteins, it's like just hydrogen bonding, and the tertiary structure has all the side chain interactions with salt, disulfide, all that stuff. Then there's also another slide on the same lecture that has a picture of like all the side chain interactions under secondary, so I wasn't sure if secondary is just hydrogen bonding, just the slides that kind of contradict themselves there, so I wasn't sure.
so. Okay. Secondary structure. Like the alpha helices and beta sheets, those are like 100% only hydrogen bonds is making those structures. But then when you're looking on the tertiary level, sorry, that was kind of confusing. Then you're looking on the tertiary level, those other, so like the hydrophobic interactions, and then the disulfide bonds, those are determining the arrangement of the alpha helices and beta sheets together. So.
Yeah, we're into the secondary structure,
Yeah.
because
Yes, and then tertiary structure is everything else, so,
and hydrogen bonds.
and hydrogen.
And more, like,
Yeah.
okay.
More hydrogen,
Yeah,
yeah.
that's.
Could you explain like the structural differences between that pregnancy and the time? I don't, there was like, I know the one had like a triple A plus thing, and then there was like a neck and a, like other.
Yeah, I mean,
Unless that's not important then.
it is semi-important, like, this is all, we can go over it now, it's in the slides. But when I asked Mackenzie about this, she basically just wants you to, I think the most important thing is to know that there's a head and a tail, and to know about the processivity, so like, the kinesins and dyneins are basically just walking along the cytoskeletal filaments. It can help to know like the more specific structures, so like, the dyneins will have that cargo binding tail, and then they have like a ring. And then they have a stop, like,
Say function more important than
yeah,
structure.
yeah, so you'll know the structure in how it is important for the function. It can help to know that like the tail is a light chain. And then everything else is a heavy chain. But it's just most important to know that the, like, what is binding to the cytoskeletal filaments and what is carrying it along. So, Any other questions?
So what did you mean with like the myosins and how they're like pulling the two positive ends? What did you mean by that?
I hate to say this again, but it's helpful to watch videos. Like when I was going, I'm sorry, when I was going through, I was like, what does it mean for example? Like, because when you look at the structure, when you look at the structure, it's I was looking at it, I was like, how can it be positive, be oriented when there's stuff on both sides? So basically, like, that would be like. Okay, so basically you would have like one side of skeletal filament here, and I might be wrong. But basically the way it's gonna work is like these heads will bind here and then they'll like pull this that way. So it's like they'll be attached to two things at a time, to two, like the time pulling them together. So it's you don't need to actually know how that works. But again, watching the video makes it make more sense. Because for me, it was just like, how is that possible in a shape like that? But like, that's like the big difference is that with the like microtubule-based motors, they're kind of like walking across the cytoskeletal filaments. But with the actin-based motors, they're pulling them together rather than walking in one direction. So it's basically just like whether they're pulling them together or like pushing them apart. This. Yeah, no, I know, this is like so annoying, but like, for almost every single unit, it's helpful to just like, that it has a mechanism, it's super helpful to just watch a video, like that's what I included in class, like, as nice as it is to do like write stuff down, like, the videos make it so that when you are reading the notes again, you can actually like visualize what's happening in your head, rather than just trying to like formulate how to explain it. Which like obviously we're not doing that great of a job at. But like that's for a reason, it's just because it's a lot more intuitive to understand. When you're actually watching it happen, and it's like one really thing rather than like this happens and then this happens. Because it's all like on the computer, Yeah, do we have any more like main content questions, structure of the past questions? Okay, thanks for coming, we will have review sessions for all of the exams, but in the future, we will probably put out or next week, like, for you guys to help me when you do that. I will have to be probably either Tuesday or Wednesday. And if you have any like personal,